Okay, so I'll get started here. So first, just a general description of what this software is designed to do. So what we're walking into the problem is we're walking in with a structure of a target molecule, a DNA protein target or something like that. And the docking software is going to take a small molecule, test a whole bunch of conformations of that, and tell you what it thinks the optimal docked conformation of that molecule is to the target. And hopefully it's also going to give you an estimate of the free energy of binding as well, so you can rank different compounds using the method. So there are a few aspects of this problem of docking a molecule, and in this little introduction I'll go through each of those. So first of all, you need a scoring function that tells you at each of your trial poses of this small molecule around your target what you think the energy of interaction there is. And so in these docking algorithms, there's a big trade-off here of coming up with a scoring function that has enough science in it to be accurate and give you a number that you believe, but still be fast enough that you can test all of the conformations you need to test to find that optimal pose. And so there's a real trade-off between speed and accuracy there. And so methods like molecular dynamics typically use a much more sophisticated free energy function, and they're going to give you much more accurate energy predictions. The docking algorithms tend to be a little sloppier, so I'll talk about that a little. How much you can trust it, I'll talk about that a little later on. The second piece of algorithm that you need for this is a search method that somehow searches the entire space of available conformations of this small molecule around the target, so that you have some expectation that you're actually finding what the best answer is. And so at the end, I'll talk a little bit about the dimensionality of that problem, which is really important when you're setting up your own experiments to decide whether you're searching exhaustively enough to get an answer that you trust. And there are problems that are way too big to be solved by these kind of methods. Okay, so a little bit of history about the AutoDock suite. I mean, there are a whole bunch of different methods these days for doing this, for solving this docking problem. And AutoDock is one of the popular methods, probably most popular because it's absolutely free. And so a little bit of the history is back in 1990, I wrote the first version in Fortran. It was the first docking method for docking flexible ligands to targets. And then over the years, we've improved this in a number of ways. First of all, by improving the scoring function so that it can predict free energies instead of just being a generic score. And we've also worked to put together graphical user interfaces to make it more and more accessible to people who aren't experts in molecular graphics. Most recently, in 2009, a postdoc, Oleg Trott, rewrote the whole program, came up with a different method of scoring, a different method of search, and came up with this version called AutoDock Vina, which is much faster, but makes more approximations than the original AutoDock. And I'll talk about which of those two are the best to use for particular problems a little later on as well. And then most recently, Stefano wrote this program Raccoon, which is the one you'll be playing with after the break, which is kind of a turnkey graphical user interface for setting up virtual screens and analyzing the results once you get them. So it's a really nice tool for that. Okay, so talking about the scoring function. So we use a scoring function that's taken directly from molecular mechanics kind of studies, but it's streamlined a little bit to make it fast enough that we can use it in the docking study. And so it has a bunch of the terms that if you've done molecular dynamics and molecular modeling, you're probably pretty familiar with all these different terms. They're based directly on the physics of the interaction between the ligand and the receptor. So I'll just walk through these one at a time. So it has a dispersion repulsion term, van der Waals term. What that's going to model is the shape complementarity of the ligand with the receptor. So in this case, this is a kinase with a nucleotide bound in there, and that's going to define this nice ligand-shaped pocket, make sure that things aren't overlapping and that it's going to find a pocket that's of appropriate shape. On top of that, we add some terms that are more specific based on the electrostatics. And so it has an electrostatics term, which in this case is going to give us this nice interaction between the phosphate and the metal ion. And there's a hydrogen bonding term, which in this case is going to give this nice specific interaction between the base and this carboxyl group. So those specific terms are giving us the specificity of recognition. On top of this, we add a desolvation term, which is important for getting the numbers right on the free energy calculation. And so what this is modeling is the kind of a whole bunch of terms all glommed into one big term, some enthalpic contributions and entropic contributions of the release of water molecules that surround the ligand and fill the binding site when the ligand binds. There'll be a bunch of water molecules that are released. So it's generally a favorable term. It also models the hydrophobicity effect, right? So that desolvation term is going to be favorable for burial of carbon-rich areas of the ligand and carbon-rich areas of the receptor. And then at the very end, we add a term to account for the entropy of freezing out all the degrees of freedom in the ligand. So this is going to, it's an unfavorable term. If your ligand is very flexible, it's going to be more unfavorable than a ligand that's a rigid body. And we use a very simple approach to that, just counting up the number of torsional degrees of freedom and weighting, giving each of those an unfavorable weight. Okay, so we add up all of those terms for each of the trial poses that we're doing during our search, and use that to find the best conformation and to estimate what the free energy is going to be. So when we were putting this force field together, we did a calibration against a bunch of known complexes where we know the conformation from crystallography and we know the experimental binding constant. Calibrated this force field by putting a little weight here in front of each of those terms, calibrating those weights so that we get the best fit of experimental energies to our calculated energies. And so here's the final answer of how well these docking softwares can do in prediction of energies. We can get it to a standard error of about 2 to 3 kcals per mole, which is not great, right? What that allows us to do is when you're doing a virtual screen of thousands and thousands of molecules, you're going to be able to tell the difference between millimolar binders, micromolar binders, nanomolar binders. But things that are different by a kcal per mole, no chance. That's all within the experimental error of our scoring function and our pose prediction. So that's a really important thing to keep in mind for how much you can trust these. If you want to get to much more accurate free energy predictions, you're going to have to go to much more sophisticated methods, free energy perturbation, all this kind of stuff, which is going to allow you to look at one system very much in detail. This docking is more for doing these huge screens and coming up with a short list of things that we think are important. So that's the AutoDock force field. We did a similar, Oleg did a similar calibration for the AutoDock Vina scoring function, which the terms are a little simpler. The functional forms of the parameters are a little simpler. He used a much larger set of 1,300 compounds for his calibration and gets about the same thing, 2 to 3 kcals. And if you look at all of the other docking softwares that are around, this is about the accuracy that people get. So, okay, so in AutoDock, another aspect of the method which we use to make it fast enough to get answers out within a few minutes to less than an hour, is that we calculate these grid maps. So what we do is take our target macromolecule and embed it in a 3-dimensional grid and then run a probe atom. So you take a carbon atom and you run it through that whole grid, calculate the energies at the beginning, and then you use that as a lookup table through your whole docking simulation. It speeds up the calculation immensely because you're not calculating those pairwise interactions between the ligand and the receptor at every single pose you're testing. So it speeds stuff up a lot. The disadvantage of it is as soon as you calculate that grid, your receptor is totally rigid, right? It can't move. So, and that's a big approximation because, as you know, proteins move a lot, induced fit, allosteric motions, and stuff like that. So that is one big limitation of these kind of methods. And we have a few kludgy ways to get around that. AutoDock allows you to pull a few of the side chains out of that grid calculation and move them explicitly during a calculation if you think a few of those amino acids are important. You can also use a method developed here at UCSD called the relaxed complex method, where you take a whole bunch of different receptor conformations, for instance, from molecular dynamics, do dockings against all of those, and then pick whichever one's the best. Right? So you're accounting for the range of motion that you'd see in that dynamic simulation. And the docking is fast enough to allow doing that. One of the big differences between the Vina force field and the AutoDock force field is the way that hydrogen bonds are treated. So Vina was designed to be very fast, so it needs it uses a gradient-based optimization when it's optimizing conformations. And that causes problems with adding directionality to the hydrogen bonds. So it uses a spherical representation of the hydrogen bonds. And so here's just some grid maps for two common types of hydrogen bond interactions. And this will give you an idea of kind of the accuracy of results that you get out of these kind of docking studies. So up here at the top, here's an arginine interacting with a carboxyl group. And so you'd kind of expect the two hydrogens of the guanidinium to form a nice interaction with the carboxyl group, just like that. The same with base pairs. You know, you want to get the nice Watson-Crick base pairing right next to each other. The spherical hydrogen bond potentials have some problems with that. So what happens is that here are the two hydrogens on the arginine group. So you put a spherically symmetric potential around that, and it turns out that it adds up and gives this big peak right between the two hydrogens instead of forming that nice lined up interaction that you'd expect. And so this is the kind of interaction that those spherical potentials will predict. If you go down, though, and make a more sophisticated potential, this is a potential that is a directional potential, saying we want to be in front of the hydrogen atom, and also that we're only going to interact with one oxygen at each hydrogen position. And then it gives results that are much more what we expect. It's much more expensive to calculate, though. With those approximations, we're not able to use to calculate the derivatives that we need to do this kind of derivative-based optimization. So the whole process is slower using that more sophisticated thing. So these are things to weigh. It turns out, for most of the problems that we're going to do, which are a biological molecule that has evolved with an active site that is evolved to bind to a ligand-shaped molecule. There are going to be a whole bunch of these interactions. And so these kind of effects kind of tend to wash out because because you're optimizing a bunch of these at once. And so Vina actually, most of the time, gives results just like what you'd expect. But if you dissect it out and try to make it do one interaction at a time, then it kind of underscores these kind of limitations of the method. Okay. Another thing that these maps are good for is for giving a feeling for some of the forces that are driving this interaction and specificity. And so we've actually used this to create a method for predicting optimal active sites within a target, which we call AutoLigand, which is also available on the site now, on the website. And so what it does is, for instance, here's HIV protease with one of these protease inhibitor drugs bound in kind of a tunnel-shaped active site. These are two of those grid maps. The white is the carbon map. The red is the oxygen affinity map. And so you can see it's kind of an image of where we think the best inhibitor would bind, or how this enzyme has evolved to recognize one particular inhibitor, or one substrate, which is a peptide coming through here and two large aromatic side chains at that center two amino acids of that substrate. And so what the AutoLigand software does is it goes into this kind of confusing map and looks for the best big continuous chunk of that affinity and says, this is what we think is the best place for building an inhibitor. This is what we think is the best guess for what the active site is for the enzyme. So that tool is available as well. Okay, so now let's jump over to the search. So again, we're going to have a trade-off here between speed and accuracy. We want to do something that's fast enough that we can get an answer out in a reasonable amount of time. So I mean, ideally, you'd want to do a systematic search. You want to take your ligand and put it every possible place and say, this place is the best. But unfortunately, for everything except for maybe a rigid body, that's far too big a space to search. So instead, we have to add some elements of randomness to that search, trying a bunch of things, maybe using gradients to push it in directions that we think are best, games like that. So what that does is it expands the range of the search method so that we can work on much larger problems, but it also means that we don't always know that we have the right answer. So we're constantly trying to validate that the search protocol that we're using is powerful enough for the system that we're wanting to answer. And I'll talk a little bit about how to assess that later on. Okay, so we have in the AutoDock suite, we have a number of different search methods that are available. The original one that I wrote was a simulated annealing method, but then very quickly after that, we moved to these kind of genetic algorithms where you create a gene, a digital representation of the variables that you're trying to optimize, and then use things like crossover and mutation to explore the range of those parameters. And so both AutoDock and Vina use kind of a really effective combination of that genetic algorithm to do the global search, and then at points during that simulation using some method to optimize the answers that we think at each time are close to what we think are the best. So in AutoDock, we use this Solus-Wets optimization, which basically just wiggles around what we think the best conformations are, looking for something that's better. And Vina actually uses the local gradient to walk down towards what is the best local conformation near where you are in the search. So those these kind of locally optimized genetic algorithms are really the best things that we have to use these days. Okay, so let's talk about the dimensionality a little bit. So we're optimizing a bunch of different parameters on this, right? We have some parameters defining where the ligand is and what its orientation is, 6 degrees of freedom there. And then however many rotatable bonds we have in it are adding additional levels of dimensionality that we have to search. And so if we just do a quick little thought experiment here, if we're just docking a water or metal ion and give ourselves 1 million energy evaluations. We want to do a simulation where we calculate an energy 1 million times. If we just start docking a water molecule, we can test 100 in each of those, 100 in X, 100 in Y, 100 in Z. That's pretty much an exhaustive search, and we're going to find the right answer, right? But now if we start adding complexity to the thing, we lose that granularity So for instance, if we have a rotatable, again, a rigid body rotatable one, then we can only make 10 evaluations in each of those. If we move to like a big peptide with 14 torsion angles, which is not unreasonable, then we can only make 2 evaluations in each of those. And, there's no chance. So things get tougher and tougher the more degrees of freedom that you add. And so here's kind of the answer of what AutoDock and Vina, based on the tests that we've done, are able to do. In terms of speed, Vina is much faster than AutoDock. So it can search a lot more. And it ends up being able to search molecules that have about 20 to 30 torsional degrees of freedom. So that's a tripeptide, maybe? Maybe a little bit bigger than that. The AutoDock, with its genetic algorithm, Lamarckian genetic algorithm, is slower, and it's down to around 6 to 12 to 16 torsions. So that's a dipeptide, maybe. Which are completely doable, if we're doing drug design. That's the size of drugs, right? We want to keep them small. We want to keep them relatively inflexible. Okay, so places where these methods really aren't suitable, the first one, the most important one, of course, is the garbage in, garbage out. You have to have a target that is effective and something that you trust is going to be close to the conformation that you think this drug is going to be binding to. We've had really mixed luck using homology models. These docking simulations are very sensitive to pretty small motions of side chains and things. So homology models need to take with a real grain of salt if you're doing these kind of docking studies with them. And then you have to make sure the size is appropriate, that the thing is not too flexible. I get people that call and say, I have an octapeptide that I want to bind to this. No way. For those kind of problems, which you want to do a lot, right? What you do is you break it into smaller pieces, dock each of the pieces, look for answers that are consistent between the things like that. You have to start playing games like that to come up with a simulation that you'll get an answer that you trust. And then, of course, another big problem is this limitation of the rigid target. If you have a lot of protein motion, induced fit, there are going to be big problems. And so, if possible, for instance, with HIV protease, which has these 2 flaps that close down around things, docking was a mess until some of the structures were solved that had inhibitors bound in the active site with the flaps closed, that were close to what we expect for the drug-bound conformation. So you always want to try to look for a target structure that's close to what you think is going to be appropriate for the small molecule you're trying in your docking simulation. And so keeping that in mind, these are some of the things that we do just to make sure that we're on track with a particular simulation. So, and as I was just saying, you want to try to, if you can, come up with a target structure that's similar to what you think is going to be the relevant conformation for your docking simulation, since there's going to be very little accommodation once you start the docking studies. And the way that you can validate that is to try to come up with a known structure. If your crystallographic structure has an inhibitor bound to it already, pop that out, do a redocking simulation just to see whether you can reproduce the experimental conformation first. Then you're pretty confident that your docking simulation is throwing enough computational energy at it to give you an answer that you'll trust, right? So those kind of redocking validation studies are very common. Another thing you can do is to perform these dockings multiple times, 10 times, 100 times, and see whether you get a consistent result across those. If you're getting 100 different answers, probably not searching exhaustively enough. If you get 20% of them, all of them coming up with the same conformation, you're probably close to the optimal. Okay, and then I guess I'll finish here with some advice about which of these two tools would be best. So Vina is very fast, and this applies to a lot of the other methods that are available, a lot of these other very fast methods. But to make it faster, it's using a lot more approximations, right? The scoring function is much simpler. So I always tell people to try the fast method first. And if you get an answer, if your redocking things are working nicely, just stay with it, you're good to go. If you're starting to have problems, then you can go to AutoDock or methods that are a little more complicated. It gives you more possibility to tweak parameters, to try adding explicit waters or who knows what, to start getting answers that are more what you expect. So also the Vina also uses this kind of map approximation to speed up the scoring function, but it does it all inside, so you never see them. The maps in AutoDock, if you want to play some games with the visualization software, they're really a nice way to get a handle for what aspects of the receptor are important for the interaction. And so I use the maps a lot just as a visualization tool, an analysis tool. So you might play with that if you have some interest in visualization as well. And I'm going to skip these two and send it over to Stefano.